Assalamualaikum everyone, this is Mariam Yusufi with matric number 1926254 and I'll be doing the introduction part of this mini project. Let's learn all about enzymes today. A living cell must carry out a large number of chemical reactions every day in order to function effectively. One issue with this is that most chemical reactions are naturally slow, necessitating the use of something to speed up the process. The use of catalyst is a preferable method to speed up a reaction. A catalyst is a substance that accelerates a reaction without being consumed in the process. Enzyme is a form of catalyst produced by living organisms. Catalyst is something that lowers the activation energy. Activation energy is the minimum amount of energy needed to start a reaction. So, the higher the activation energy, the more energy is needed to start a reaction, resulting in a slower chemical reaction. However, enzymes lower this activation energy, so the reaction that takes place in our body can happen trillion times faster than it normally is. Enzymes can work in different ways. Some enzymes help to break down large molecules into smaller molecules. Others build large molecules from the smaller ones. But how do enzymes do that? Enzymes work in conjunction with the substrate. The enzyme and the substrate are always moving and occasionally collide at the right speed and orientation. The substrate sits into the enzyme at a region called active site. Active site is a region within enzyme that fits the shape of the substrate molecule. From this, we can tell enzymes are highly specific. We can explain the way enzymes work using a lock and a key mechanism, where a special molecule is the key to activate the enzyme function. Once the substrate is in the active site, the reaction takes place and the required product is produced. Enzymes are sensitive molecules because their activity does not only depend on cofactors but also on the surrounding environment. Some of the major factors that influence enzyme functionality and activeness are temperature, pH, enzyme concentration, and substrate concentration. In this mini lab project, we will be focusing on the temperature factor. Imagine having an enzyme catalyzed reaction and gradually increasing the temperature. At each temperature, the activity of the enzyme is measured and a graph like this is obtained. As you can see, as we increase the temperature, the activity of the enzyme increases. In other words, the reaction gets faster. At a certain temperature, the enzyme is working at the fastest possible rate. That's called the optimum temperature. For most human enzymes, the optimum temperature is 37 degrees Celsius. As we increase the temperature past the optimum, the activity of the enzyme rapidly decreases to zero. In another words, the enzyme stops working, and the active site is denatured. The objective of our experiment is to determine the effect of the temperature factor on the enzyme's activity. In this experiment, we will be measuring the enzyme activity at 3 different temperatures. To do this, an experimental setup is established with the help of yeast and flour. The flour is first mixed with yeasted water at 3 different temperatures, 28, 37, and 100 degrees Celsius. The yeast works as an enzyme in the experiment. As we know, yeast ferments sugar and starch present in the flour and produces carbon dioxide and alcohol. This results in different dose sizes. The dose sizes are then measured for each temperature to determine the temperature effect. The manipulated variable in this experiment is the variation in temperature. The control variables are the amount of water, flour, and the yeast use, and the responding variables can be measured by comparing the dose size either visually or by the use of the measurement tools.
The material prepared for the experiment is a bowl, cling wrap, instant yeast as an enzyme, flour as a substrate, measuring tape, thermometer, a glass, spoons, water heater, and lastly plastic gloves. Now I'll talk about the procedure. First, put 3 tablespoons of flour into a bowl. Second, dissolve a teaspoon of instant yeast into the glass and stir it with 28 degrees Celsius of water, that is water at room temperature. Third, pour the mixture of the yeast into the bowl of flour. Fourth, mix the mixture with flour until it's fully mixed with. Fifth, wrap the bowl with cling wrap. 6. Start the timer and set for 15 minutes. 7. Measure the early side of the dough and then take the measurement of the dough until after 15 minutes. The procedure is repeated with different temperature which is with 37 degrees Celsius and 100 degrees Celsius to get the desired result on effect of temperature on enzyme activity. Now I'll talk for the result. For water in 28 degrees Celsius, the radius of the dough increased from 6 cm to 8 cm. There is an increase of 2 cm in the dough. For the optimal temperature of 37 degrees Celsius for water, the dough radius increased from 6 cm to 8.4 cm. There is 2.4 cm difference. And lastly, for the water at 100 degrees Celsius, that is boiling temperature of water, there is no change in the radius of the dough. At higher temperature, the enzyme will denature and the reaction cannot be occurred. That's all for this experiment.
Assalamualaikum warahmatullahi wabarakatuh. My name is Jahan Azri binti Yaka Ali, matrix number 1921738, and I'm in charge of result and discussions. Here, we can see the results of our group experiment presented in a table. The time taken and initial diameter is same for all three temperature, 50 minutes and 6 cm respectively. The final temperature for 28 degrees Celsius experiment is 8 cm. For 37 degrees Celsius is 8.4 cm, and for 100 degrees Celsius is 6.1 cm. The diameter difference, delta D, is calculated by deducting final diameter and initial diameter. For 28 degrees Celsius is 2 cm, for 37 degrees Celsius is 2.4 cm, and for 100 degrees Celsius is 0.1 cm. Before we analyze the results, let me explain some theories that will help in understanding the results. By increasing the temperature of substances or compounds, more energy will be supplied that will lead to increase in the particles' kinetic energy, and more collision will happen among the particles as shown in the animation at bottom left of the screen. Same goes to enzyme and substrate collisions. The more collision happen between enzyme's active site and substrate, the enzymatic reaction will increase. However, enzymatic reaction will only increase until it reaches the optimum temperature of that particular enzyme. Optimum temperature is the temperature that an enzyme shows its highest activity. If it's in a graph, it's the peak of the graph. Due to enzyme characteristic that is very sensitive to temperature, the enzymatic reaction drops after it reaches optimum temperature. Higher temperature will cause the enzyme to denature. Changes in enzyme active site structure causes it to be unable to bind to substrate anymore, as we can see in the animation at bottom right of the screen. Now, let's analyze the result using graph. As you can see, the diameter difference, which represents the enzymatic reaction, increased from 28 degrees Celsius to 37 degrees Celsius. However, it drops after it reaches 37 degrees Celsius and keep dropping until 100 degrees Celsius. Maltase is the enzyme that can be found in yeast that is used to break down maltose into glucose. Glucose is a type of sugar. So it will be consumed by yeast for fermentation process, and the process will produce carbon dioxide and ethanol. So the release of carbon dioxide gas influences the difference in diameter of the dough. The ethanol that released by the process will be burned out when the bread is baked. The more degradation of glucose happens, the more carbon dioxide released by yeast, which results in higher diameter difference of the dough. Theoretical optimum temperature for the maltase is 37 degrees Celsius and proven to be correct in this experiment. The peak of the graph belongs to 37 degrees Celsius. As I mentioned before, peak of the graph indicates the highest enzymatic activity. Increase in temperature afterwards causes decrease in the enzymatic reaction of the maltase, which we can see the diameter difference drops. The highest temperature, which is 100 degrees Celsius, results in lowest difference in diameter, 0.1 centimeter, which also can be taken as the lowest enzymatic reaction. At this point, the maltase will denature, and there won't be any difference in diameter if the experiment proceeds with an even higher temperature. Hence, temperature does have an effect on the enzyme activities and also enzyme kinetics. Now, let me relate enzyme kinetics and bioinformatics. Bioinformatics is a field that develops method and software tools for understanding biological data. It also can used to collect data, store data, analyze data, and other features. Enzyme kinetics information is very important to develop medicine and clinical diagnosis. Some bioinformatic databases can be used to obtain enzyme kinetics data and stimulate enzyme kinetics by providing a kinetic rate model. For example, WRK, as seen in the picture here, and Brenda are bioinformatic databases. Copacy, on the other hand, is an enzyme stimulator. The stimulation by Copacy can be used to predict reaction time, derive equivalent concentration of substrate and product, and many other data. Here is a picture of Brenda homepage. Here, by using Brenda website, I obtained data about the enzyme used in this experiment, which is maltase. Various information can be obtained once we click search. As can be seen in the left side of the screen, there are few headings such as enzyme nomenclature, enzyme ligand interactions, functional parameters, organism-related information, enzyme structure, molecular properties, application, and other information. Some of the information are synonym terms which can be found under enzyme nomenclature. So maltase is also known as Saccharomyces cerevisiae. Under the functional parameters, we can find the Km value and Ki value listed here. pH optimum and also temperature optimum is also listed under the functional parameter heading. Maltase pH optimum is 6.6 while its temperature optimum is 37 degrees Celsius. By choosing the enzyme structure headings, we can obtain the amino acid sequences of maltase as shown here. Since maltase is a type of an enzyme is a protein as well, so it has its own amino acid sequences. There are many other information and data can be obtained from Brenda databases just by searching the name of the desired enzyme. This is the picture of a Copacy application. So development in collecting, storing, and analyzing information in bioinformatics field will be able to develop further the uses of enzyme in biotechnology and biochemical field. That's all from me. Thank you.
To conclude this mini project, some important findings are highlighted. As expected, the final dot diameter at 37 degrees Celsius is the largest. This is because enzymes work best at their optimum temperature, which in this case is 37 degrees Celsius. At 28 degrees Celsius, the dot size increased as the temperature increased. However, the enzyme activity dropped as the temperature rises above the optimum temperature, resulting in a lower dot diameter size at 100 degrees Celsius compared to both 37 degrees Celsius and 28 degrees Celsius. Significance and applications of enzymes. Enzymes are found everywhere in the industry. Some applications of enzyme in industry are pharmaceutical industry. They're used in making pills and drugs. Food industry. Enzymes are required for the production of cheese, yogurt, wine, and so on. Environmental management. Enzymes are powerful tools to help sustain a clean environment in several ways. They are utilized for the environmental purposes in a number of industries such as agro-food, oil, detergent, and petroleum. This concludes our mini project. Thank you.